In this week's lab, we will be isolating chloroplasts from plant cells. So you may be wondering, why chloroplasts? First of all, plants are important, even if you're going to medical school. Second, chloroplasts are really easy to observe using light microscope because they are green and have a lot of contrast. It was really important for early scientists to be able to isolate organelles from cells, and that's because cells contain many different kinds of structures, molecules, and organelles. In order to initially understand what each of these organelles are for, they had to separate them out from everything else in the cell. This can be achieved using a technique called differential centrifugation. To start a differential centrifugation experiment, we first collect tissue. So in our tube, you can see that we have a lot of animal cells, okay, but obviously other types of cells are valid too, including the plant cells that we'll be working with. The next step is to homogenize these cells, which means to lyse or rupture them, so that their cytoplasmic contents will mix together kind of like a soup. Once these guys are homogenized, we're ready to start the centrifugation. Some things in the homogenate are larger and have more mass than others. For example, nuclei are heavier than mitochondria, which are heavier than ribosomes. So we'll be able to sort these out by mass. To separate these organelles by mass, we'll use an instrument called a centrifuge. The centrifuge will spin samples at very high speeds. This speed creates force that pulls molecules and organelles to the bottom of the tube in proportion to their mass. We'll see an example of that on the next slide. Heavier items are going to be condensed into what's called a pellet, or a little bit more solid structure, at the bottom of the tube, and things that are still lighter will stay in the liquid above. This is called the supernatant. It's easy to remember that the supernatant is the liquid on top if you think about it as soup like the food. You can perform successive spins at increasing speeds and times to isolate what you need. So let's look at an example. Taking our homogenized cells from the previous steps, we can take these guys for a spin. Starting relatively slow, we can spin at 1,000 times gravity for 10 minutes. And yes, that's Earth's gravity. Once we've finished this spin, the nuclei and larger structures should be pulled to the bottom of the tube, while things that are a little bit lighter will stay in the liquid. After this spin, we can do a couple of things, okay? Take our supernatant and our nuclei. If we wanted to work with the nuclei, all we have to do is remove the supernatant and put it into another tube, and then we have a pellet that will consist of mostly nuclei. If we want something even smaller, we can discard the nuclei and take the supernatant to the next step. So in this spin, we did 1,000 times gravity for 10 minutes, but to get smaller and smaller items, we'll have to speed it up for longer. Our second spin is 20,000 times gravity for 20 minutes. After this step, things that are about the size of a mitochondrion will be pulled to the bottom of the tube, while things that have less mass will remain in the liquid supernatant. So we have again our supernatant, and here we have mitochondria. If we wanted the mitochondria, we simply remove the supernatant and take the pellet, which will be composed of mitochondria at this point, and do whatever we need to with it. If we wanted something smaller, we'll again take the supernatant to the next step, spinning even faster and for longer. The last step that we'll see today is 80,000 times gravity for an hour. In this stage, we'll be able to collect ribosomes, and there actually are things that are smaller in the supernatant that we can mess around with if we wanted to. So we could either take the ribosomes as they are, or take smaller things that would be like small pieces of lipids, tiny pieces of membranes, even smaller proteins, those types of things. So we can actually get even smaller. So this is probably enough for now to understand at least the concept of what we'll be doing. So now that we know how differential centrifugation is performed, we're ready to start the second video. In that video, you'll actually be able to see the protocol that we'll be using in lab to isolate chloroplasts specifically.